very much for the kind introduction and for also inviting me to give us a chance to promote this the method we have developed on the site. So before I start, I just want to mention that most of the stuff was done by Pokia Chen, who was a postdoc in my lab. He's now back in, so you can see my screen, I hope, right? I mean, my presentation. All right. So Pokia Chen was really instrumental in this entire endeavor. So he had the basic idea and did most of the measurements and came up with algorithms, etc. And all the pipeline we presented today is also downloadable. You can check out in the papers, in the published papers, the link where you can download from GitHub the entire pipeline. So first I want to tell you how we actually got the idea to utilize small X-ray scattering for ligand screening. I mean, screening in SAXS is of course a normal thing. It's 96 well-paid format due to the great beamlines. Synchrotrons, they've installed there all the automation and so on, very nice. But to our knowledge, at that time at least, wasn't really used to determine affinities. And we thought it might be good because what we usually do is we are interested in the complex life of RNA. This is the central dogma of molecular biology, where DNA is transcribed into mRNA by the polymerase, then translated by the ribosome to proteins, of course, and we have all the intermediate steps, 3' end processing, splicing, etc. And we are especially interested in the parts here of the general RNA binding proteins, regulate transcription together with long non-coding RNAs, or also regulate translation. And this we do, as already mentioned, in an integrated structural modeling approach. Just as a summary what this is, so let's assume you have a typical RNA binding protein, which is often modular. So often you have 4 RNA binding domains, like RNA recognition motors, for example, connected with often flexible linker regions. And let's say the 2 central ones, they really bind RNA, and you might have input structures determined by X-ray crystallography or NMR. And then you have information about the RNA binding interface of these proteins. And then you can, of course, determine a lot of other restraints. This complex might be too big for NMR, so you can use sparse NMR data, cross-linking mass spectrometry, small X-ray and Newton splitting, of course, EPR, FRET, and other methods. And then you can use, during structure calculation, simultaneously these restraints to obtain a final ensemble. So this is what I would call the simultaneous approach. Of course, you can also have the filtering approach where you first, based on the structures you have, you let allow during structure calculation and desimulations the linkers to move around. So you get a purely MD-based ensemble, which tries to cover the theoretically potential conformational space. And then you use all the restraints to filter against your models, and you maybe come up with a final ensemble which is similar to the other one. And then, of course, you can mix this also, that you use some restraints for simultaneous structure calculation, and the other restraints you can use them as validation against your ensemble. Of course, you might also be lucky to obtain a crystal structure or cryoEM structure of this entire complex. Then you could use all the restraints to validate this, the complex in solution, which actually might be quite useful sometimes. If it's the same, all is good. However, you might realize that there's actually some dynamics, and this then you can refine the model, the solution model, basically. The problem is we have in this field is that when you want to determine the protein RNA complex structure, is that you need to know the right RNA to which the protein binds to. Because if the RNA, for example, is too long, you might have sliding of your complex, of your protein on the RNA, which prevents crystallization, which gives rise to line browning in NMR. You can, it might be too small, the RNA, so that only one RNA binding protein binds. Or the sequence is not optimal, that you have also intermediate exchange behavior, which also is bad for crystallography and also bad for NMR and cryoEM also, of course. So you need to identify the right RNA motive, whether it's a structural motive or a sequence motive. And there are several methods to determine RNA specificity of RNA binding proteins. There are a lot of in vivo methods which are quite powerful, but not easy to use. And you need sequencing, for example, here RNA immune precipitation followed by sequencing, where you pull down RNAs bound to your RNA protein, RNA binding protein. So you have a tagged RNA binding protein cells, you pull it down, and then you do sequencing. And from the pulled down RNAs, you can look for similar features and you might get a motif. Better is cross-linking immunoprecipitation, where you're crossing the RNA to your RNA binding protein. So in this way, you can even identify the approximate site where the protein is bound on the RNA. So the motif might be more reliable. You have tried an RNA tagging, etc. And then you have high throughput in vitro methods like SAXS, RNA compete, and RNA bind and seq, which are also very powerful. The problem is also that you have often a maximum 8 to 9 nucleotides identified, which is good for tandem RNA binding protein, tandem domains, but not more because the sequence gets longer. And then, of course, the difference between in vivo methods and in vitro methods is, I would say for structure determination, if your aim is structure determination of a complex, the in vitro methods are better because you have a very defined system. The system you test, you want to determine structure of. In vivo is very interesting because you have maybe some additional information because there are all the other RNA binding proteins available, which maybe change the specificity of your protein or of your complex towards a certain RNA. So the motif you get out from the in vivo methods might not be the one which is optimal for structure determination. Nevertheless, if from both methods you would then refine the motif using biophysical experiments like scaffold independent analysis, NMR, ITC, Biocore, EMSA, switch sense, and so on. And now we thought, since these methods either use quite a lot of material or are low throughput, like ITC and NMR, or you don't get directly additional structural information. So we thought maybe to test small X-ray scattering as a tool to screen for the best RNA. So the beginning is to discuss about RNA binding proteins, and later on we tried also to, I will show you some other data to apply this also onto small molecules and receptor ligand situation. So we call it SAXS screen in the end, and just as a summary, small X-ray scattering, I guess most of you know, however, maybe it's good to say nevertheless for those people who have no experience with small X-ray scattering. So the same, just in short, is about the same principle as X-ray crystallography. You have an incoming or neutron, of course, neutron or X-ray beam, but you don't have a crystal, so they're not aligned in ordered fashion, your molecules, but they tumble freely in solution isotropically, or more or less isotropically. So that's why you don't get a clear diffraction pattern, but you get a scattering intensity, different scattering intensities dependent on the angle. So you plot your intensities versus scattering angle, and you get a curve like this here. So at low angles, you get strong scattering intensity, where you get information about the global properties, like the radius of gyration or the molecular weight. And at wider angles, you get structural details. So sometimes people show this, but it's not really true. You cannot really talk about the resolution. So the linear dimension is 10 angstrom to 1000 angstrom. Of course, you get only a shape of the molecule. So you can determine oligomeric state of your complex, the shape or conformational changes, interaction of different macromolecules, and of course, the variations of points 1 to 3 as a function of pH, salts, and ligands. So when you add ligands, you might have a conformational change, and this you can screen for. You can use also contrast variations, so you can even visualize within a larger complex individual subunits. This is better neutron scattering, but it's less sensitive, of course. So what's important is that your total scattering intensity you get is always a sum of the individual scattering intensities of your components. So if you have a mixture, then this intensity, of course, is a mixture of all the, or it's a sum of all the intensities of your components in your sample. This is important to note. So the idea behind this is if you have a receptor which binds to a ligand, you have a conformational change, and this can be differentiated by small X-ray scattering. It needs to be large enough, the conformational change, that you can see it by small X-ray scattering. And this is something you need to test, or you know by experience approximately, you can imagine that this should be able to monitor by small X-ray scattering. And then you can extract populations from the total average. And then from this, you can determine affinities. So then we first needed to think about a good model system. If you want to develop a method or some kind of algorithm or pipeline, of course, you want to test this on a good model system or also to improve it on a model system. And of course, we have plenty of those. We decided to take the protein SAXS. It's a female-specific protein in Drosophila, where it's involved in dose compensation, one of our main biological questions in the lab. It has two Rn domains, RNA recognition motifs 1 and 2, which binds to RNA. So from the crystal structure we determined in 2014, also has been determined before in 1999 without the protein UNR in complex. We know the specificity for RNA. So we know that, for example, in this position, it's important to have a G, purine specificity to a guanine, and the yellow ones are specific for a U, a uridine, and actually the white ones, it says no preference, but there's also a U preferred because the pockets are better for a smaller pyrimidine base and a purine base. We also have the crystal structure from the paper 99 of 3,6-lethal, and there you could see that the domains are a bit more extended from each other because the linker is flexible, which you could also see in NMR, that the linker residues here, if you look at the rotational correlation times, that the linkers have lower R2 over R1 values, so it's flexible, allowing the domains to move away from another. As a summary, so in APO6-lethal, domains are a bit extended, they can move with respect to each other. And when you add RNA, they're getting more compact, and the radius of gyration, for example, would change because the void in the middle is filled by RNA. Okay, the problem was, so we thought this is a good model system, and our initial SAXS experiments actually were a bit surprising because we could see that the RG didn't change as much as we would expect from theoretical calculations. And what we realized when we looked at it by NMR was that when we have in dark blue, we have the 6-lethal wild type. If you look, for example, at this peak here, 6-lethal wild type, so both domains together. And in red, it's only the RM1 domain, and in yellow, it's only RM2 domain. So there seems to be interaction between both domains, also in absence of RNA. When we increase the linker length by including some GS repeats, GSS repeats, 10, 20, and 25, we can actually get also the tandem domain construct towards the single domains, meaning that we can disrupt the interaction by increasing the linker length. So in the end, we use 6-lethal with a 10 GS linker included into the system. And this is now, we can theoretically back calculate the curves from the from ensemble of the three 6-lethal structures, which is like this one here. And you see then if we add RNA, we should expect a change of the SAXS curve, that you can really have a different change. And then you can plot the RG values, make linear fit of this, and also volume of correlation, another parameter which takes entire curve into account, introduced by John Tyner a while ago. And so we thought we should be able to actually determine the affinities from this. And this actual data, initial data, so you see this is the RNA alone in green. In black is 6-lethal alone, the 6-lethal 10GS variant. And then in red is the experimental curve when we had both 6-lethal and G10. And if we would only add up the green one and the black curve, we would come actually to the yellow one. So you wouldn't see much of a difference. So indeed, we have a change in curvature, which we, in the same way, we more or less predicted also. When we use C10, so this is U10, quite close to the optimal sequence, and this is C10. This we introduce with an offset to see it better. It's the same curvature, basically the same curve, so we have no binding. So this we can easily see between a good RNA ligand and a bad RNA ligand. Okay, so now we have, this is a pipeline basically. So we now do a 96 well plate. We have 12 times 8, so we have 8 different RNAs in one plate, and then like 10 titration points, a buffer in the end and a buffer in the beginning. So often in SAXS you measure buffer sample, but then you would decrease the throughput quite a bit because we tested this on several synchrotrons, and it's quite stable if you only measure the buffer in the beginning of a titration row. and in the end of the titration. And then what you do, so again, you have the basically total scattering intensity, and this is of course the mixture when you have RNA of a free state and the bound state. But with a linear minimization, you can actually get then the bound fraction out of this out of your measurement, and then this you can use for quantification also. So you can subtract basically the intensity of the free receptor, of the free ligand, and of the buffers, and so on, and then you can basically get the population of the bound fraction. Okay, and this is then what you get. Here are 3 different RNAs. You do the processing, and then you have 3 different parameters: the radius of gyration, volume of correlation, and the chi-linearity we introduced. And you can actually see that it looks quite like binding curves. Again, yellow C10 does not bind. UG-U8 seems to bind better than U10, which we would also expect. And this is also then you can make a pairwise reduced chi-fit matrix also here. And then you can plot basically relative KD, where UG-U8 has the strongest affinity, and A10 and C10 are quite weak. Okay, so these are the overall results. So we tested all these kinds of different RNAs, and you can see if the RNAs are too short, for example. So the longer they get, from U5 also to U10, the stronger is the binding. And UG-U8 is indeed the optimal RNA binder. So it's the strongest affinity. A10 is not so good, C10 is not so good, etc. When we compare with ITC, we have actually a 10 times higher affinity for UG-U8 in ITC than for SAXS. These are quite okay, do all right. And U10, we have actually 1 times lower affinity than in ITC. The problem what we had here was that dependent on U10 and UG-U8 sequences, we have quite a different sample. And SAXS gives you actually this information for free when you do a clustering. So we have for certain RNAs a clear 1-to-1 stoichiometry. For U10, for example, we have 94% in the 1-to-1 population. But it could be, according to our MD simulations we used to fit the SAXS data, we could be in a situation like this. So that's why this would be quite weak binding, because only 1 domain binds, or this. Then we have these low affinity RNA binders, and we have a 2-to-2 stoichiometry. And this was especially for UG-U8 here, where we have a scenario where you have a bit of aggregation, you have 2-to-2 populations, etc. So it's a mixture. So that's why we think that the affinities deviated quite a bit from ITC. Okay, but we were quite happy that we could use this method to identify the best RNA binder. Again, so this is now a pipeline. This is all available from GitHub, as you can see, and yeah, 96 well-paid. And then we have the SAXS titration profiles, and we can directly get all the parameters: radius of gyration, volume of correlation, or this chi-lin. And which one you take actually depends a bit on your system. So often the radius of gyration, which would be the easiest one to get, can be enough if you have a change from a really more extended conformation to a compact conformation, because then it's already in the beginning of your curve, when you do the linear approximation. Otherwise, it might be better to use the chi-lin or the volume of correlation, because it takes the entire curve into account. And we will introduce another parameter later on, as I will show you, because the next step we thought would be interesting to see how well this pipeline would perform with the receptor small ligand interaction. Again, so you have what's usually used, and we heard our talks today, is of course crystallography to identify best binders, fragments, and so on, and then to extend the fragments. NMR, of course, is also good. You get at least residue resolution, but you need quite a bit of materials. cryoEM is nowadays also possible to do this, but it uses quite a lot of optimization. It's not really possible in high throughput. Crystallography, yes, once you optimize the entire system, but you need quite a bit of material, of course. And then, of course, you have all the biophysical experiments. So we wanted to also see if we can include SAXS into this pipeline, the same principle as with the RNA. And we actually contacted Novartis, for example, if they would be interested. The scientists were interested to test this on their system, but then the legal department said, no, they don't want to send any samples. So we looked at the literature and thought that this might be a good test system here: periplasmic binding proteins, which when they bind, for example, small amino acid binding proteins and so on, there's a small conformational change from the free state here on the left to the bound state, and you see like a clamp closes a bit. So the practical radius of gyration change is only 1 to 3 angstrom, and the overall shape is actually quite similar for SAXS. So it's a low resolution method, but so we want to test if this is actually possible to see this change and to get affinities from this. Again, these are theoretical curves between both states, so differences are not too big, as you can see. So we also wondered what concentrations or signal to noise ratios do we need to precisely predict the affinity. So high enough concentration to get good signal to noise ratios to see the difference between both curves. And then we also want to see, if they're across synchrotrons, if it's then reproducible, which we expect it to be. And these are literature values from different amino acid binding proteins. The histidine binding protein is quite nice, has a wide range of affinities. The strongest, of course, for histidine, 64 nanomolar, and the weakest for ornithine, 72 micromolar. These ones we tested, I think, these two also, glutamic and aspartic acid binding protein and glutamine binding protein, and there you have an even wider range of affinities. And this is some of the data. So on the left is for histidine binding protein. Again, 96 well plate formats, so 8 different amino acids and around 10 titration points for each step. And they all look very similar, but if you would zoom in, if you look a bit closer, you see here a change for histidine. The gray from gray to green, it starts from no ligand to excess of ligand. Also for lysine and arginine, you see the same. And then for the other ones, there's almost no change in the curvature. This is for glutamic and aspartic acid binding protein, where you also see a change. So if you zoom in, there's quite some change, and the metric definitions we use here now is again the chi-lin, so how well the composite curve can be fitted by linear combination of the curve of its components. The radius of gyration here in the Guinea range gives you information about the average 1D mass distribution relative to the molecular center of the mass, and then Poker introduced the volatility ratio. Comes from the financial sciences. It's a change in rate of fluctuations in intensity between two curves per Shannon channel. So you divide basically your curve into Shannon channels, and then you get this kind of volatility ratio. You can all read it in the papers. The theory behind it is described there. And now this is what we got. So 3 different parameters. And here, for example, for the volatility ratio, you can see that you actually get quite nice binding curves. They look like binding curves, of course, a bit scattered. For the radius of gyration, not so much, because this one doesn't change so much. It's very small conformational change, so this was not very reliable. But the chi-lin and the volatility ratio worked quite well. And this is how it compares with literature. So we also measured ITC ourselves as a control, and here are literature values. And for histidine binding proteins, so histidine is clearly the strongest binder, 28 nanomolar plus minus 32, the error is quite large. And then ITC gives us 45 and literature 64 nanomolar. So it's quite in the same order of magnitude at least. Same for actually arginine. So it's 2.4, 2.3, and 3 micromolar for arginine for our volatility ratio, SAXS experiment, our ITC experiment, literature values. And then for weaker binders, we have a bit higher deviation. For lysine, it's still okay. But then for ornithine, where you have almost no change, then of course it's very noisy. So the difference is very small, and then it's fitted to a very high KD. Okay, so as a summary, so this is a pipeline here, 96 well plate format. You can imagine this as that you have a free state here and the bound state, and here for the blue one, the stronger binding, you have more proteins fully bound, and here where you still have some free fraction in a certain ratio. Again, this you can see here, zoom in also that you have these difference in the SAXS curve from which you then can determine affinities. Okay, so I mentioned always high throughput. So compared to cell-based assays and so on, of course, it's far away from high throughput. The major problem we have at the moment in throughput is actually the washing of the capillary after each measurement. So the measurement takes actually only 2 to 10 seconds, depending again on your concentration. So we actually use what I forgot to say. I think it was on one slide mentioned. So we used 2 mg per mil, around 50 micromolar. I think we could have gone down to 0.5 mg per mil. We tested. It also worked quite well for histidine binding protein, at least. And each measurement takes 2 to 10 seconds. But then the buffer, the capillary wash, takes 1 to 2 minutes. So this really increases the measurement time. But when I was measuring at Diamond, for example, the local contact showed me a very nice like drum-based system or revolver-based system where you had 5 holders and 1 cylinder. And then while one is measuring, then it's done, it turns, measures the next, and while this one measures, it washes the other ones. So this would be a solution, for example, to improve the throughput quite considerably. And then you could measure, I don't know, a couple of 100 ligands per day or per session. So there are room for improvements, and of course you have to think, I mean, we're basically done with this. We thought it was quite fun and interesting to do, and we might use this RNA binding identification of best RNA binders. We use SAXS screen sometimes, but I don't know, it depends really on the system. It can be very useful for certain systems where you can get easily good data. I mean, the conformational change needs to be large enough, basically. Okay, with this, again, thanks to Poker, and of course, we measured this across 4 synchrotrons. We basically got the same results on each of them, all via remote measurements, so there was no problem in handling and so on. We sent the 96 well plates, just had to spin down them, and that was it. Okay, thank you. That's it.